Good morning to everyone present here. I hope you're all having a pleasant day so far. I am Sharanya Dumre Patil and I will be your host for the day. It is with great privilege and pleasure that we extend a warm welcome on behalf of Amity University Mumbai to its distinguished lecture series on the biology of tuberculosis. Today's lecture will be delivered by the esteemed speaker, Dr. Deepak Kaushal sir, and the topic is non-primate preclinical model of tuberculosis, its infection and comorbidities. The series is organized by Amity Institute of Biotechnology. Tuberculosis is one of the deadliest infectious diseases with 1.5 million global deaths and 3 million fresh cases in 2021. In the relentless pursuit of understanding and conquering tuberculosis, the utilization of non-human primate model has emerged as a crucial avenue of research. These models provide a window into the complexities of tuberculosis, offering an insight into not only the disease itself, but also its interaction with co-infections and comorbidities. The knowledge gained from these investigations hold the potential to revolutionize our Okay, so these investigations hold the potential to revolutionize our approach to diagnosis, treatment, and prevention, ultimately shaping the landscape of global health. As we embark on this intellectual journey led by distinguished expert in the field, we are presented with the unique opportunity to peer into the intricacies of tuberculosis research. This lecture promises to unveil the remarkable interplay between tuberculosis and other factors that impact its manifestation, progression, shedding light on the potential avenues of intervention and breakthroughs. That said, I would like to introduce Dr. Venna Suprasanna sir. He was the former head of Nuclear Agriculture and Biotechnology Division BARC Mumbai, former professor at Homi Bhabha Institute Mumbai, India, and has joined Amity University Mumbai as Dean of Research, Professor, and Director of Amity Center for Nuclear Biology, Biotechnology and Head of Institute of Amity Institute of Biotechnology. Now I request respected Dr. Venna Suprasanna sir to share the welcome address.
Thank you, Sharanya. Good morning to everyone, and a warm welcome to Professor Deepak Kaushalji. We are very happy to extend welcome to Dr. Deepak Kaushal. He is one of the top researchers in mycobacterium and made fundamental discoveries on tuberculosis, the way the pathogen infects, the way infection proceeds, and all the mechanistic insights into the biology of tuberculosis. I think we are very fortunate to have Professor Deepak to tell us more about the biology. Again, a warm welcome to you. I'm very happy that AAB is organizing the biology of tuberculosis lecture series, because this opens up new window to the tuberculosis, the way the pathogen invades our human body, and the way infection spreads, and what kind of treatment methodologies are available. I think this is a very good initiative, and all thanks to Dr. Shashank for initiating this, because he came up with the idea, then we worked on that, and then he could talk to eminent experts like Professor Deepak to talk on this. Thank you, and warm welcome to Deepak ji. Thank you, Sharanya.
Thank you so much, sir. We are so glad that you took time out of your busy schedule to be part of this event and gave us an amazing welcome address. And this brings us to the main part of the lecture. I would now like to invite Dr. Shashank Kamble, sir, the Assistant Professor at AIB, to introduce it.
So good morning, all of you. It is my privilege to introduce Dr. Deepak Kaushal. I had a brief interaction with him when I was in JNU in a conference, and he's such a nice person, so down to earth. So to introduce him, Dr. Deepak Kaushal did his PhD from University of Delhi South Campus. After that, he had a brief, he had postdoctoral experiences at John Hopkins School of Medicine at Baltimore, and along with that, he had a visiting fellowship at the Institute of Genomics Research at Rockville, MD. Dr. Deepak Kaushal is currently working in the in the Texas Biomedical Center as the director for the National Primate Research Center, which is a part of Texas Biomedical Center. He's a principal investigator of 15 NIH funded grants and is a co-investigator of 9 NIH grants. He's also funded by the Bill and Melinda Gates Foundation among the private granting organizations. His lab mainly works on using the MAC model of TB using the natural inhalation route of infection, and which is also a model for the MTB HIV co-infection. So he has done extensive research and made so many breakthrough discoveries about how MTB interacts with the primate lungs and how it forms granuloma, and this has provided fundamental insights into the biology of the pathogen as well as the host environment, and the establishment of a robust MAC model of inhalation for TB and HIV/AIDS goes to him because he has established this platform for so long. And this can be used as a novel vaccine candidate also, because you can also test novel vaccine candidates in such kind of MAC models, and you can study how the drugs works against TB in a live infection model. So thank you so much, Dr. Deepak Kaushal. Now I hand over the stage to you.
Thank you all. Thank you, Sharanya. Thank you, Dr. Suprasannaji, and for your kind words, and particularly thank you, Shashank, for inviting me to give a talk here. I actually owe Shashank. The last time I met him at that conference, I made him wait, which was not something that I want to ever do to anybody. And so the least I could do was accept this invitation and talk about my work. So I'm going to share my screen, and kindly let me know if you can hear me well and if you can see my first slide. So I'm going to talk about pretty much the only thing I know, which is the non-human primate model of TB, and how we use it in my lab to understand fundamental aspects of this deadly disease, which impacts the whole world, but particularly India, and also how to make discoveries that would be transformative in the field of TB treatment and prevention of TB, therefore therapeutics and vaccines. I work at and currently direct the Southwest National Primate Research Center. As Shashank mentioned, it's part of an independent institute, the Texas Biomed Institute. The SNPRC is one of the 7 national primate centers in the country. I earlier worked at Tulane University, and they have one of the other 7. And much of the work that I'll be presenting today, Tulane was my first ever faculty appointment. I started there as an assistant professor in 2006 and moved here to direct this center in 2019, just before the pandemic. But 13 years of my first 13 years of my facultyship were spent at Tulane, and therefore much of the data that I'll be presenting today was actually generated there. I've been fortunate enough to work with great trainees, both PhD students, postdocs, residents, et cetera, and I'll try to acknowledge them as much as possible. And I'm not trying to tell you one story today. I'm taking tidbits from different studies to just have people understand what this model does and can offer. I'm very happy to note that there is a growing understanding of the role that preclinical models like non-human primates, monkeys, can play in India as well. Just last year, I got contacted by the ICMR, and there was a joint ICMR NIH National Institutes of Health program that sent one veterinarian from an institute in Hyderabad to my lab, to my center, so that he could learn how to manage monkey colonies and how to do different procedures and so on and so forth. And he got here in February. He's actually leaving tomorrow. So he spent exactly 6 months here on a J1 visa, and I hope that he will take some of this knowledge back. So that's great. I don't think, for this audience, I need to spend a lot of time talking about the importance of tuberculosis research. It's something that still devastates population in many parts of the world, including India. But just to put tuberculosis, the disease, in a clinical perspective, it is caused by a gram-positive bacillus called Mycobacterium tuberculosis, which is spread by aerosol. In fact, TB is one particular pathogen where we have documented irrefutable proof that it spreads by aerosol. So people who have it in their lungs, cough, talk, sneeze, and expel micro droplets that contain these bacteria that can be inhaled by those near them. And this is how up to 2 billion people, it is estimated, have gotten exposed to these bacteria and have productive responses. A very small number of, a small percentage of these individuals, believed to be between 5 to 10%, actually develop disease. But because the pool is so big, the small percentage equals about 10 million cases of disease every year worldwide, and between 1 to 2 million people dying from TB, and the majority of cases and the majority of deaths due to tuberculosis actually occur in India. And there are heterogeneous and non-specific symptoms, clinical symptoms of disease that are listed there. And this is the reason why I chose to work on tuberculosis, molecular aspects of tuberculosis during my PhD and postdoc, as Shashank mentioned, and have continued to work with this disease in my independent career. So my lab primarily uses animal models, non-human primates, monkeys, to study the granuloma. That's one program within the lab. The granuloma is a pathological structure, and we're trying to show you here as part of a cartoon that one of my former PhD students, in fact, my last student at the Tulane University, Ali Bukshan, made this cartoon to try and explain how, in very simple terms, how TB works. So the pathogen, Mycobacterium tuberculosis, is inhaled by naive individuals, and it goes into the airways of individuals, and there are airway lining alveolar macrophages that pick up this particular bacteria. So here I'm trying to show you an alveolar cartoon of an alveolar macrophage that has been productively infected. And these cells make, in response to infection, a lot of chemokines and cytokines that attract other cells. Initially granulocytes, neutrophils, basophils, etc. And these cells are loaded with oxidative stress armament. So they try to mount oxidative stress to try and eliminate the pathogen. And so this concept of oxidative stress early on in TB is very important. Please keep it in mind. I'll return to it. When there is a failure by the lung airway immune system to eliminate the pathogen, then B cells and T cells, lymphocytes are recruited to the site of infection, and this creates an initial and intact granuloma. Granuloma is essentially a pathological structure that defines TB. There are granulomas in absence of TB. You can get granulomas in some other conditions, but there is no TB without granulomas. So if there's productive MTB infection in a person, and there is clinical disease, then there must be at least one granuloma. Over the course of time, the center of this granuloma caseates. There is poor vascularization because the blood vessels cannot go into this pathological structure that's walled off. And so the center of this granuloma becomes hypoxic or has low oxygen concentration. And this is another very important concept that I'll return to later. The disease spreads when bacterial growth is not stopped inside of a granuloma, and these granulomas break open, thereby causing both intrapulmonary and extrapulmonary spread of the disease. So that, in very simplistic terms, is how TB works. 15 years ago, when I got recruited to a university that had a national primate center, by that I mean one of the 7 NIH, National Institutes of Health of the US, supported primate centers that are just standalone facilities to try and develop models of human disease and ailment. Then I decided to build a monkey model of TB using the natural route of infection. So there were some people that had tried to infect monkeys before I became an independent faculty, but they had tried to surgically instill TB bacilli into the lungs of monkeys. And when I was recruited as faculty, I then recruited a brilliant aerobiology postdoc who's now become an independent professor. And together we worked to develop this model where monkeys are anesthetized and then they're placed in dorsal recumbents in a head-only chamber. Inside this head-only chamber is an AGI, all-glass impinger and nebulizer that creates aerosols. So we throw in tuberculosis solution, and by controlling the concentration of tuberculosis bacteria, bacilli, in the solution and the amount of time that this particular monkey is being exposed to these aerosols that are being produced right here in front of you in this impinger, and also by controlling the breathing rate of this monkey, we can determine how many bacilli go in and what kind of infection occurs in these animals. The beauty of the system is that this is a class 3 environment. So you can see this is a glove box. So people operate this from the outside, so there's no exposure. So you could be wearing street clothes like this and still operate in this facility and expose monkeys to TB. And so that's what my lab literally does every time. Now, why did we decide to develop a monkey model of TB, which one of my first postdocs, Dr. Smriti Mehra, who's also now an independent professor, helped me create. And the reason would become very apparent with this slide that I'm showing you. So as I mentioned to you earlier, when people get exposed to TB, billions of them do not get any disease. And a small minority, 5 to 10%, actually get disease. And we cannot mimic that disparate, very polar phenotype in any other model. If you use mice or other rodents, then you get you only get progression. You don't get latent control of tuberculosis infection. But as you can see here, we can get that in monkeys. So I'm showing you lungs from 2 different monkeys that were infected the same day at the same time with the same dose of the same strain of Mycobacterium tuberculosis. And one monkey, the one on the left of the screen, ended up sick soon after infection, within months of infection. And we had to euthanize it because it had reached all its endpoint criteria. And when we euthanized the animals, we saw, as you can probably see here, lobe-level consolidation and hundreds, if not thousands, of tuberculous granulomas. The other monkey, which I mentioned to you, infected the same day, same dose, same strain of TB, had no other signs, no signs of disease. So we kept it alive for the duration that the animal protocol said we should keep it alive. It had tuberculin skin test reactivity, but no signs of disease by blood measurements, by radiology, by any other means. And when we euthanized this animal, we could see just a handful of small granulomas that did not have any bacteria in them. So this monkey on your right represents those billions of people that get exposed, that get coughed upon and get exposed, say, in the Bombay local train, but do not have any disease. And the monkey on the left are those millions of people around the world that actually progress and cough and spread more disease and will die if they're not treated with antibiotics. And so this is the central reason why I chose to develop this model. And nowadays, we don't have to actually euthanize animals to look at this disparate polar nature of disease in the lungs, because we can do whole body PET CT imaging, and so we can recapitulate that without actually having to euthanize animals, as you can see in this particular slide. This is a technique developed in my lab by postdoc Bindu Singh, who was earlier a PhD student at the National Institute of Immunology in Delhi. And it's not just gross pathology that's important in these animals. When you look at the histopathology of the granulomas that we get out of these monkeys, they look very much like human granulomas. Whereas when you infect mice with aerosols of TB, you don't get that architecture that is central, that is germane to how human tuberculosis progresses, as you can see here. So what you can see on the left is there is a central caseous region of the granuloma, followed by a middle lighter colored ring of cells, which are granulocytes and macrophages, and then a darker ring of dark blue cells, which are smaller cells, B cells and T cells. So as I showed you initially, that's how the granuloma forms. First, there is infected cells and oxidative stress, then macrophages, and then T and B cells. And you can see that very clearly in the macaque granuloma slide, as opposed to the mouse slide. And we've belabored this point further. So Dr. Mehra in my lab ended up showing that in our monkeys, we can see all kinds of pathology presentation in these monkey granulomas. They're not just caseous granulomas, but they actually exhibit fibrosis, mineralization or calcification, giant cell formation, which is typically associated with bad TB, cavity formation, which is also bad TB. That's how the bacillus escapes into the bacilli escape into the airway, and post-primary lesions. And she actually developed specific techniques like RNA-scope to actually identify that these granulomas have live bacilli and produce Mycobacterium tuberculosis specific RNA in these bacilli. And then I was fortunate enough to have a board certified veterinary anatomical pathologist and histopathologist, Dene Lobato, come to my lab. She didn't think she had enough degrees, so she wanted a PhD in my lab. And she ended up showing even more specific pathologies of tuberculosis, such as septal thickening, type 2 hyperplasia of the lung, perivasculitis, and lymphangitis in the monkeys. So your all kinds of human TB presentation, even some rare ones, can be seen in our monkeys. So you get the drift. The point I'm trying to make is that these monkeys, even though they're experimentally infected, their lungs look like humans that have TB or humans that have controlled TB. And then in her PhD work, another former PhD student, Terry Haddock, ended up showing that these monkeys are actually hypoxic. These granulomas in these monkeys are actually hypoxic. So she developed a stain that would go into any part of the tissue, animal tissue or human tissue, and we call it hypoxy probe. And if the regions were hypoxic, then it would stain. And you can see the granulomas actually do stain hypoxic, and the genes that respond to hypoxia are actually turned on in these monkey lesions. I'm not showing you data that this does not, as you could imagine, happen in mouse granulomas. And then another former postdoc, Maya, ended up showing the obvious result that in those monkeys where there is progression of disease, so remember my first lung slide, there was a lot of bad TB in one of the animals. Those animals not only have a lot of pathology, but they have a lot of bacteria growing out of those lesions, whereas those that control TB have very little pathology and very little bacteria. And those blood markers that we would associate with inflammation and tissue damage, such as serum C-reactive protein or serum AG ratio, so your very clinical markers, can actually be recapitulated just like you would expect in this preclinical model. My lab specializes in single cell level analysis of granulomas in these TB infected animal models. So we were the first to develop single cell RNA sequencing and single cell site-off methods to study TB in these granulomas. So we have three types of monkeys. We have monkeys that have progressed to active or pulmonary TB. We have animals that have stopped that progression. So very few granulomas, we call them LTBI monkeys. And sometimes we have to use controls that have not been infected. But we perform generally deep unbiased analysis using single cell techniques in my lab, and then validate using conventional techniques like flow cytometry and imaging. And in very elegant work that Katya Isalova and Dheeraj Singh, Dheeraj is a former PhD student of JNU and a former colleague of Shashank's, they did was to identify the single cell transcriptome of TB granulomas in these monkeys. And this was published in a very good journal a couple of years ago. And they established that the reason why there is inflammation in tuberculosis is because there's an influx of specific interferon responsive interstitial macrophages, CD163 positive, CD206 or MRC1 negative macrophages that bring all this inflammation. We have other ways of doing single cell analysis of the granuloma. This technique that I'll describe next is called Mibitoff. We do consecutive cycles of labeling our granulomas or lung slides with antibodies that are labeled with different metal ions. So we can do up to 37 cycles. So you can imagine you can take 37 different markers that you want to study and label them with different metals and then stain. And then these stains are carved out of the slide and put into a mass spectrometer, a time of flight mass spectrometer. So essentially we are taking the microscope and mass spec and bringing them together. And then the computer software actually provides us pseudo images that are single cell level resolution and recreates the granuloma. And we can ask where it, and in one part of the granuloma, what particular gene or what particular protein is being expressed and what pathway is being expressed. And this is work of a brilliant shared PhD student, Erin McCaffrey, from Stanford University. So I think what I've shown you thus far is that macaque that MTB infection in our monkey model, particularly in their lungs, recapitulates key aspects of TB disease. But another important question is, what do the granulomas of these monkeys and therefore presumably humans, which our monkeys seem to recapitulate, do to TB? And we've tried to answer this question over the last 10 or so years by doing transposon screens. So what we do is we take a transposon and insert it into a TB genome. And we've modified this transposon so that it can only jump once. So if it goes into a particular gene and interrupts it, it cannot jump any further. And that transposon is barcoded and has an antibiotic selection. So then we get a stable mutation in that particular gene. And if we do this experiment, if we do this transposition event again and again, we can cover pretty much the whole genome of tuberculosis that has 4,000 genes. This was a technique that was developed in the lab of Bill Bishai, Professor Bill Bishai, a noted TB clinician in whose lab I postdoced at Johns Hopkins almost 2 decades ago now. And so we started running screens of the type I'm showing you in monkeys and mice in parallel in my lab. And what we found was that if we took 100 mutants and pooled them together and threw them into monkeys and threw the same pool into mice, very few mutants were lost in mouse lungs. And a lot of them were lost in monkey lungs. So the implication is that the genes in which the interruption is important for growth in those lungs. So what that means is a large number of these genes are required for growth in monkey lungs, and very few of these genes are required by MTB to grow in mouse lungs. And here I can show you a little bit more data because we ran the screen again and again. The key data point is that almost 1/3, 33% of all the genes that we could interrupt in the in the TB genome were required for granuloma formation in the monkey lung. Whereas in comparison, only about 6% of the genes were required for formation of granulomas in the mouse lung. So the implication is that the monkey lung, presumably the human lung, and the granulomas that form in that environment put a lot more stress on Mycobacterium tuberculosis than do other models. And that should not be a surprise to you based on what I've shown you thus far. I'll belabor this point with two different examples. So DOS-R is a transcription factor that is encoded by the tuberculosis genome. So what is known is that when you show Mycobacterium tuberculosis hypoxia, you take away oxygen concentration, even when it's growing in a culture in vitro, then DOS-R is induced. It's a transcription factor, and it's induced through two different sensor kinases, DOS-S and DOS-T, and then it induces the expression of a number of genes which presumably help Mycobacterium tuberculosis survive against hypoxia, which happens inside of granulomas. You may remember I talked about it earlier. Now, Dave Sherman from Seattle discovered this gene and did some of the really early elegant experiments. And he showed the beautiful biochemistry of this pathway. But when he made the mutations in this pathway, so Mycobacterium tuberculosis lacking DOS-S, DOS-T, or DOS-R, he could not find any phenotype in mice. The mice, the bacteria grew just fine in those mice, just like they were Mycobacterium tuberculosis. So we took his strains and we infected monkeys with it. And you can see the legend on the right. So we infected with either the whole wild type Mycobacterium tuberculosis or the DOS-R mutant. Remember, DOS-R is the transcription factor, or the DOS-S or the DOS-T individual mutants. Those are the two kinases that signal and talk to DOS-R and induce it. And then we also had the DOS-S, DOS-T double mutant because we just wanted to make sure that the signal couldn't go through one or the other. And then we had a complemented strain where DOS-R had been knocked out and then put back in a translocation. And we infected groups of monkeys. And as you can see here, initially infection is established more or less the same, but at a time when granulomas form, so 3 weeks is a time when granulomas have not formed, 7 weeks post-infection is a time when granulomas form. And you can see all the DOS mutants are controlled. The only place where bacteria grow in the lungs are MTB, which is the wild type isogenic strain, or the complementary strain where the gene has been put back again. And the same is true at a later time point. The same is true for pathology. You only see pathology in the virulent strains, not in the DOS strains. And the same is true for killing. The survival is there in all the DOS mutants. Now, these mutants grew just fine in mice and killed mice, just like Mycobacterium tuberculosis does. But in monkeys, you can see a very distinct phenotype that is revealed. The second example is that of sigma H. Sigma factors are, sigma H is an alternate sigma factor, one of the many that is encoded by this bacterium. And my own postdoctoral work captured in a PNAS paper about 20 years ago actually showed that this particular sigma factor responds to oxidative stress. So again, clinically very important. Remember the initial granuloma cartoon that I showed you. Bacteria must first solve oxidative stress before it moves on and granulomas are formed. So what we discovered was that sigma H induces the expression of thioredoxins, and these thioredoxins are really important genes for Mycobacterium tuberculosis because they can scavenge oxidative burst through this chemical reaction that I'm showing you. So as the host granulocytes and macrophages mount oxidative burst, that happens through molecules like hydrogen peroxide, as you can see on your screen, or superoxide ion. And these thioredoxins can reduce those harmful chemicals to unharmful chemicals through this chemical reaction. And that's why induction of thioredoxins is important for mycobacteria. So you can imagine a situation, think of this as a macrophage vacuole or phagosome. Bacteria goes in, there are enzymes like iNOS and FOX that make all of these superoxide ions or peroxide. And if you have thioredoxins inside of this vacuole being released by Mycobacterium Bacterium tuberculosis. Then this stress gets scavenged, bacteria survive, they can replicate, they can form granulomas, and disease happens. Now, when I infected mice with the sigma H mutant, there was no phenotype. The bacteria grew just fine, just like wild type. But many years later, when Dr. Mehra infected monkeys with the sigma H mutant, there was a day and night different phenotype, while those monkeys that had wild type Mycobacterium tuberculosis infection progressed. Those that had delta sig H did not show any signs of infection. And as you can see here, there's 100,000-fold difference in the bacterial levels in those lungs. So from these exercises, we conclude that macaque TB granulomas mount both a greater magnitude of and broader stress. So stress is broader, therefore you have hypoxia, and you can see those phenotypes revealed. The magnitude is greater, therefore you have phenotypes like SIG-H revealed. Now, this work allows us to go in to vaccine discovery. There are different kinds of vaccines that are tested by various postdocs and grad students in my lab. But the one that has become very interesting for us is this attenuated MTB delta SIG-H mutant that I just talked about a second ago. And why? So the sigma H mutant is not attenuated in mice, but it is attenuated, I just showed you, in non-human primate lungs. We found it's also attenuated in non-human primate, excuse me, and human macrophages. So if you just take macrophages out of a human lung through bronchoalveolar lavage or a monkey and infect them with wild type MTB, so CDC1551 here is a clinical isolate of MTB, then MTB can grow in those macrophages because it's an obligate pathogen of macrophages. But look at what delta SIG-H does. It's controlled, just like BCG vaccine. So here's an indication that the strain could potentially be a whole cell live vaccine. Now, when we do RNA sequencing of these macrophages, we see that those macrophages that are infected with delta SIG-H mutant actually show a hyperimmune phenotype. So they express chemokine cytokine genes that are involved in protective immunity, and they also express markers of trained immunity. So again, giving us an indication that this could be a very good vaccine strain. So we did experiments with delta SIG-H mutant to study why we were getting a hyperimmune and trained immunity phenotype with this mutant. And we thought it could be because delta SIG-H fails to inhibit phagolysomal fusion. Now, TB owes its success as a pathogen because it successfully inhibits phagolysomal fusion. So when the bacteria go into the macrophage tries to fuse its phagosomes and lysosomes. Lysosomes have low pH and oxidative stress kills bacteria. Mycobacterium tuberculosis says, no, let's stop that process so that we can thrive and we can persist. So the assay for phagolysomal fusion is the overlap between the green color, which is the bacteria, and the red color, which is LC3, a marker for phagolysomal fusion. And as you can see here in our ex vivo assays, sigma H actually has much higher levels of phagolysomal fusion than bacteria. So this was a very interesting result. And what could be the consequences of failing to inhibit phagolysomal fusion? That would be greater antigen presentation, because when the bacteria is entrapped into phagolysome, just like it appears that sigma H is, then it gets chopped up by the phagolysome, and then peptides of its proteins can be presented to the immune system, getting you both CD4 and CD8 T cell responses, your classical 101 immunology. And so we devised an assay to study this. So we did an antigen presentation assay. This is a really tough assay to do. We did an antigen presentation assay ex vivo. So we took bacteria, either TB or the sigma H mutant. We took macrophages, we fed them bacteria. We had to have antigen presenting cells in there, so autologous dendritic cells from these donors. And then we had a T cell hybridoma that could only present a specific epitope of MTB that's present in both MTB and the mutant. And if there is a successful antigen presentation, then we can read that through a classical IL-2 type secretion. And as you can see here, delta SIGH presents much more, is much more efficient at presenting antigen than the wild type. And this is your classical phagolysomal fusion driven process mechanism, because when you inhibit with siRNA for Beclin-1, then that sigma H phenotype goes away. But when you use a scrambled siRNA, that phenotype remains. So all of these ex vivo assays were telling me that sigma H can be a very good vaccine. And so we've done these vaccine studies now in human beings. And you can see here, those monkeys that are vaccinated with BCG or gold standard have a little bit less bacteria in their lungs as compared to unvaccinated animals. But then those monkeys that have been vaccinated with delta SIGH actually have significantly lower bacterial burdens. And when we go into the monkeys and look at the total number of lung lobes or the total number of samples that we've derived from these lobes that are sterile, then you can see here unvaccinated animals, almost 80% of the animals we can grow some sort of bacteria out of. With BCG, this number reduces to about 60%, and with sigma H, this number reduces to about 15%. So there's a significant effect. Andrew Lackner was the person who first recruited me to a faculty position. He was a clinical pathologist. He used to tell me, Deepak, don't show me graphs, show me pictures. A picture is worth 1,000 words. So here I'm showing you pictures. For clarity, I have put black arrows around where the granulomas are. And as you can see here, unvaccinated infected animals, hundreds of granulomas with BCG, there's an effect. There's reduction in number of granulomas, so that's a good thing. But with our test vaccine, huge reduction in the number of granulomas. We've now gone in to do flow cytometry, antigen-specific flow cytometry, to find the mechanisms of why delta SIGH is inducing stronger responses and protecting. And we find out that delta SIGH actually induces strong CD8 responses. These are CD8 responses to Mycobacterium tuberculosis cell wall. So we have overlapping peptides, so we can span the entire TB immunome and find out which particular antigen or combination of antigens delta SIGH is inducing. And again, I showed you my lab was one of the first to adopt the use of single cell RNA sequencing techniques to study what's happening. And as you can see here, we can take the airways of these animals that are either not vaccinated or BCG vaccinated or vaccinated with our test vaccine and find out what are the different clusters of lymphocytes that are being recruited and which are being recruited significantly more in our vaccination as compared to gold standard vaccination. And then we can identify the genes and the proteins that are being expressed by these clusters. So therefore, your true single cell level correlates of protection have been identified here. Now, there's a Geneva consensus that says that, you know, if you want to use a whole TB strain as a vaccine against tuberculosis, then you must have more than one mutation, and these mutations must be independent and stable. And there's good reason for it. You know, you don't want the vaccine strain to revert to wild type and cause disease in people. And so Garima Arora, who's a postdoc right now in my lab, she's a former PhD student of Dr. Ramandeep Singh in THSTI Faridabad, has come into my lab a couple of years ago and very energetically made mutations in the delta SIGH background. So we've married SIGH mutation with these other mutations. These are all rational mutations. They're all well thought out. So these are independent of SIGH. There's no interaction between these genes and SIGH gene and so on. And there are several other criteria, and these are now being tested in monkeys. And will probably be ready for human testing sometime next year. And this is a paper that's in preparation. This paper can actually go out pretty quickly, but Dheeraj's paper, the vaccine study paper that I showed you with the single mutant, is currently stuck in a high-level journal, and they have demands of us. So it might take us 6 months to actually complete that paper, and then we'll send this paper out. And after that, we will be looking for a clinical collaborator, either in South Africa or in India, to unveil some of these double and triple mutants that we've made using these basic science experiments. So just to change gears and talk about some co-infection and some comorbidity, primarily co-infection. The TB HIV syndemic is a real one. A number of people, particularly in the continent of Africa, that have latent tuberculosis. In other words, they have competent immune systems and control the infection, with initial infection with tuberculosis. Once they reach adulthood and become sexually active and become HIV positive, then they do actually get a resurgence or reactivation of TB. Conversely, many individuals, infants, babies who are born in Africa to HIV positive mothers and therefore get HIV through the mother's milk, once they start going to school, acquire TB, and they also have a much faster progression to TB because of the previous presence of HIV. So think of this endemic in just these two lines here. If you get infected with TB and you are completely immunocompetent, there might be a 10% lifetime risk of developing TB. But if you have HIV co-infection involved, then that increases to a 10% yearly risk. So if you are 16-year-old, sexually active, LTBI TB infected, HIV, you get HIV infected at the age of 16 in Africa, then by the time you're 26, you would have had a 100% chance of having developed TB. In many of these cases in Africa, this is multi-drug resistant TB, therefore huge clinical complications. Our monkey model can help. We can recapitulate this endemic by infecting monkeys with MTB and by mimicking HIV infection using simian immunodeficiency virus, which is a monkey-specific virus that's very similar to HIV. And in fact, some people believe that HIV originated from SIV2, one of the two simian immunodeficiency viruses, and we use that virus as a surrogate. So many years ago, my lab developed these co-infection models. So our model is we do low-dose MTB infection. Those animals mostly stay latent, as shown by this green bar. And when we co-infect these animals with SIV challenge, then a majority of those that are co-infected actually develop disease and have to be euthanized. And the initial, so this slide actually tells you that co-infection can be modeled in our monkeys. The initial dogma for co-infection in humans was, oh, TB is contained within granulomas, and they are coated on the outside by lymphocytes. And HIV, what it does is human immunodeficiency virus is trophic for CD4 lymphocytes. It comes in and kills all those lymphocytes, removes them from the outside of the granulomas, and then these granulomas become leaky and bacteria can escape, and therefore you have this reactivation. And my former PhD student, Ali Bukshan, challenged this hypothesis, and in a very elegant paper published a couple of years ago in the Journal of Clinical Investigation actually showed that this was not the case. So she took monkeys that had latent TB and had been infected with SIV, and then she took some monkeys that were not infected with SIV, but she depleted CD4 cells to the same level as what SIV does by using an antibody. And she had some controls there. So that antibody is called CD4R1. It's a simianized antibody that only depletes monkey CD4 cells. And she did a lot of titration so that she could get the depletion rates equal. And then she used a control virus, which does not deplete CD4 cells. And what she showed was that you get markers of disease only in those animals that are infected with the virulent or the pathogenic SIV. not with the non-pathogenic SIV and not with antibody. So you can deplete the same level of CD4 cells from the granulomas of these monkeys, but you don't get reactivation. And this is consistent with clinical markers, PET CT scans, pathology, and you do not get disease in these animals. As you can see here, the disease levels with the antibody are significantly lower than those with SIV. So she concluded that it's actually chronic immune activation caused by SIV or HIV and not the mere depletion of T cells that causes activation. We've since gone on to develop this model further. So now we can do antiretroviral therapy in these monkeys. So here, monkeys are infected by aerosol with TB. They're co-infected with SIV, and then we do the same level of antiretroviral therapy that a human would get today, especially in the US if they were to come positive with HIV. And we can do both ART and we can do TB therapy, and we can do just TB therapy, ART. I'm not going to show you data because of lack of time, but we can mimic all of this stuff. And this is primarily work by Riti Sharan, who was a postdoc in my lab and is now an independent professor. So coupling these different therapeutics, these different therapies with PET scans, we are now primed, because we can recapitulate what happens in humans, we are now primed to test new therapeutics, and we're actually doing that. And that's what the slide says. So I will stop here and thank all the brilliant people that have been in the past in my lab that are listed here in italics, and my current staff and my collaborators and my funders, primarily the National Institutes of Health, Allergy and Infectious Disease, and then direct funding from the National Institutes of Health, Office of Director, funding for the vaccine work by Bill and Melinda Gates Foundation, and a few local grants to a few postdocs. I'll be happy to take questions. I'd also like you to know that we are recruiting postdocs. So if you know somebody who wants to postdoc in my lab and work with a challenging pathogen, then they're welcome. We are in San Antonio. It's in the South Texas part of US. The top three pictures are actually views from my home. This is a very beautiful hilly terrain. And San Antonio is a very historic city. The bottom three pictures are from the downtown. It's known for its Alamo and its it's a party city, and for this Party Central River Walk. I'll be very happy to take questions at this point of time.
So thank you so much, sir, for a nice talk. I have a couple of questions. So while using the non-human primate model for tuberculosis, have you observed any dormant M tuberculosis in the mesenchymal stem cells in the bone marrow?
Yes. So actually, one of your former colleagues, Anu, is now a postdoc from. So a number of Shashank's former colleagues ended up in my lab. They were all PhD students in the lab of Dr. Gobardhan Das at JNU. Incidentally, Dr. Das is now director of ICER Bhopal, as of today, I'm told. So she's doing those experiments in the lab. She did those kinds of experiments in Dr. Das's lab as well. I don't know the answer to that question, but if there's one particular model that can answer that question, definitively, I personally believe it is this model.
Yeah. So one more question is that it's a little same thing. So you said you showed that the delta SIGH mutant basically has reduced burden in the lungs of the infected mice. So in that case also, it will be interesting to study the dormant TB in the bone marrow, specifically in the mesenchymal stem cells. And the last question is that so we have so many vaccine candidates against Mycobacterium tuberculosis, but the disease like tuberculosis also exists in other mycobacterial. It's also caused by other mycobacterial species, collectively called as MTBC complex. So, is it possible to design or a kind of a pan MTBC vaccine candidate, rather than like as you showed that the mice lungs and the primate lungs, they respond differently to the mutants, transposal mutants. So just like that, because of because of the difference in the physiology, is it possible to design a kind of pan MTBC vaccine which will work in all the hosts?
Yeah, it's a very interesting question. I think the field is gravitating a little bit away from that concept, though. I think we have to recognize the heterogeneity, both genetic, people in South Africa versus people in India versus China versus, let's say, the US or Europe, the Caucasian population, the microbiome, the prevalent co-infections, and just the way T cells of these different genetic backgrounds attack the pathogen. There are huge heterogeneity, huge layers of heterogeneity involved here. And then TB is not just one disease. There's the primary active disease, the HIV-induced disease. I didn't have the time to talk about comorbidity with diabetes, type 2 diabetes, which is big in India. And so these are all different diseases. So I think we actually need more like 5 or 10 different strong vaccines against TB. And one might work in the HIV population, but will be an absolute no-no in other populations. One might work in immunocompetent population, but we will probably not risk it in the HIV population. Some that may work in some genetic or geographical backgrounds. So I think we can't have enough specific vaccines. Maybe we need one for the infants and one for adolescents and adults and so on and so forth. Those are my
in the same line, we talk about M. bovis, which infects cattle, M. marina, which infects fish pathogens. We have M. avium, which infects birds. So just like that, all these MTBC pathogens, which infects different hosts, maybe animals, humans also. So is it possible to design a single vaccine which will work against all of this bacteria in different animal host?
Yeah, I think it's unlikely, right? I mean, yes, bovis and tuberculosis are close enough that they may have shared T cell epitopes, but marinum and avium are highly, divergent in terms of their evolution. So I think we will struggle to do that.
Okay, so thank you so much. So others who has a question, they can ask. If no questions are there, then we can end the meeting. So I don't think there is any question, sir. Thank you so much
Wonderful.
for being present for this talk. It's our pleasure indeed, and we would like to hear you again.
The pleasure is all mine. I'll actually be in India for the World Cup in 2 months' time.
Okay.
I'll be very happy to meet you then.
Sure. Thank you, sir. Thank you so much. Yeah.
Take care. Bye-bye.